Welcome to our first screencast for Chapter 11. What we're going to do in this chapter is we are going to look at the protozoans. Now when we think about protozoans, we think about very tiny one-cell creatures. If you look down here at the bottom, we have something called unicellular eukaryotes. Now the prefix uni that you see right here simply means one. Alright, so we're going to be looking at one-cell creatures. But before we start talking about our protozoans, what we need to do is we need to establish some evidence of life. Now, life first occurred some 3.5 billion years ago. Now, the first life that we were looking at during that time would have been prokaryotic bacteria cells. Now, if you remember back in biology, we had talked about two different groups of cells. We had talked about prokaryotic cells, would be cells like bacteria, and eukaryotic cells, which would be the cells that make up our body. The way that these cells, or these eukaryotic cells, evolved was through a process called symbiogenesis. Right? Now, the prefix symbio sort of implies that we have a relationship that occurs between two different organisms. Now, there's actually three different types of symbiogenesis. The first one sort of gets us started in the process. Now, again, 3.5 billion years ago, we had prokaryotic cells. And so in order to get a more complicated cell, we needed to have one prokaryotic cell actually engulf another prokaryotic cell. So what happens here is that one prokaryotic cell that's taken in the other, that second prokaryotic cell sort of becomes one of the structures found within that previous cell. So that's sort of the first step in terms of eukaryotic cell evolution. The next step would have been what we consider primary symbiogenesis. Excuse me. This would be where the primitive eukaryotic cell, so the one that resulted from the first type of symbiogenesis, actually engulfs another prokaryotic cell. And so again, we're sort of adding to the number of structures we would find within that cell. So that's considered primary symbiogenesis. The third type is going to be called secondary symbiogenesis. Now this is where we have now a somewhat complicated eukaryotic cell actually engulfing maybe another eukaryotic cell. And this is actually the way that mitochondrion and maybe some other plastids formed in more complicated eukaryotic cells. And if you look on the right here, you can sort of see how this primary and secondary symbiogenesis would have occurred. And in this case, we're looking at the formation of the mitochondrion. Up here you're going to notice we have the host cell, which is going to be that first somewhat primitive eukaryotic cell that is working to consume its first prokaryotic cell. So if you notice, the bacterium, which is going to be the prokaryotic cell, is being engulfed by that first cell. Once it's been engulfed, it's going to form a membrane around that bacterium. And so that bacterium, over a period of time, is going to evolve or change into something that that cell can use. And in this case, it would have been the mitochondrion. And that mitochondrion is important because it actually provides useful cell energy to do several different types of processes that happen throughout the cell's lives. And so that's a little bit of information on how the evolution of eukaryotic cells might have occurred. So what we want to do next is we want to go ahead and start discussing the phyla that we're looking at in Chapter 11, which is going to be the protozoan. Now we need to look at the actual word itself, protozoan, and look at this part right up here, the word zoa. Now zoa means absence of cell wall, or at least one motile stage. Now motile means movement. So there needs to be at least one stage in the protozoan's life cycle where it's going to be able to move from place to place. And we're going to look at locomotion in a second. Now, some of the characteristics of protozoans that you see listed here sort of place them into this rather unique group. First off, protozoans are extremely adaptable. In other words, you can find them in lots and lots of different types of environments. Of course, they do require moisture. You know, they have that very thin cell membrane. They are one-cell creatures, so moisture is important, but they can be found on land, they can be found in the water, they could be found on this desk right now. As long as the moisture is there at some point in their life cycle, they have what they need. But they do require moisture. They can be sessile. Now sessile means basically they could stay put, so they don't really have a lot of movement when it comes down to their life cycle, or they can be free swimming. We're going to talk about protozoans that actually have either cilia or flagella. So those are the ones that actually can move a bit more freely through their environment. Now they do definitely have what we consider an enormous role in the economy of nature. Now, they do lots and lots of different things, but the two primary ones is that they form a lot of the substrate that you would find in oceans. Now, when we say substrate, we're talking about the stuff that would make up the bottom of the ocean. And we're going to talk about a couple in a second that will sort of illustrate that idea. Of course, food. There's a lot of creatures in the ocean, a lot of creatures in several types of freshwater environments that definitely do use protozoans as a food source. Now, these particular creatures can form what we consider unique relationships with other organisms. So there are some out there that basically stick to themselves. They don't interact with other organisms too much, but there are some that actually do benefit from relationships with other organisms. Now, there's three different categories when it comes down to these relationships. We have what we call mutualistic, commensalistic, and parasitic. Now, a mutualistic relationship is where both partners benefit. The example we could give of that would be the trichonympha in termites. This is a one-celled protozoan that actually lives in the gut of the termite, and its main job is to help digest the wood that the termite takes into its body. Commensalistic is where one benefits and the other is not harmed. And then, of course, the one that we tend to be familiar with is parasitic. So this is where the protozoan will get inside or on the organism, and the protozoan benefits, but the other definitely does not. It may not die immediately, which would be not too beneficial to the parasite, to the protozoan, but over a period of time it does become weakened, then eventually in the end it will probably die. So let's go ahead and move on to our next slide. We need to look at defining some protozoan groups. Now, the first way that scientists actually did define protozoan groups was through the type of protozoan they were. In other words, what could they do to actually obtain their nutrients? We broke them down into two categories. We talked about the plant-like protist and we talked about the animal-like protist. Now, in this class for zoology, we're not going to focus on the plant-like protist too much. Our main focus is going to be the animal-like protist. But we do need to be familiar with how to distinguish between the two groups. If you are a plant-like protist, you would be considered autotrophic, which means that you synthesize your own food. Now, synthesize means to make your own food. The ones that would do this would be the ones that can photosynthesize. They use the energy from the sun to convert it into a chemical energy that the organism can use. Now, the ones that we're going to look at are considered the animal-like protist, and they're considered heterotrophic. They obtain their food from other organisms, and so these are the organisms that actually have synthesized certain types of molecules that these protists can actually take into their body. So they actually have to go out and they have to consume other organisms. Now, there's two different ways that they can actually do this. We have some protozoans that are considered what we call holozoic feeders, and some that are considered saprozoic feeders. Now, if you're a holozoic feeder, you're going to perform what we call phagocytosis, and this basically means that you are going to consume particulate types of food. So this would be very small little chunks of food that would be floating through your environment. If you are a saprozoic feeder, you're going to be considered an osmotroph, and this means that you're going to consume soluble food, which means that the food itself has actually dissolved into the environment, maybe into the water, and it's going to be more sort of like a diffusion type of feeding. So the two different types of heterotrophic feeding mechanisms would be phagocytosis and osmotrophs, and I do need you to be able to distinguish between the two, so that is very important. Now, it says down here, it says the distinction based on food is somewhat unclear. Now, as I said before, in the past, we had actually distinguished between protozoans based on their nutrition, in other words, how they obtain their food, whether they were plant-like or whether they were animal-like. Now that we've obtained more information on these protozoans, on these different types of protists, we're coming to find that actually there are many out there that can actually switch back and forth between their feeding behaviors, and the euglena would be a really good example of that. If you put a euglena into an environment where there is plenty of sunlight, it can actually become sort of autotrophic. It can actually produce its own food. It has chloroplasts, which are the organelles within the cell, that can actually take in the sunlight and convert it over to chemical energy. But if you take that euglena and the environment for whatever reason becomes devoid of light, so the sun is now gone, it can actually switch to more of a heterotrophic type of feeding behavior and can actually go out and consume other organisms. So because we have more protists that are like this, it's not as easy to put them into either a plant-like or an animal-like protist group. So when it comes down to defining, again, protozoans, in addition to looking at feeding behavior, we also need to look at locomotion. As we said before in the past, there were other ways that were used to place these organisms into these groups. Locomotion was another one. Again, because we've learned more, scientists don't use this one quite as much, but we still look at it anyhow. The three primary groups of protozoans in regards to locomotion would be considered the flagellates. We have a group called the ciliates, and we also have a group called the amoebas. Now, if you're a flagellate, that means that you use one or a few long flagella. So you would have a protozoan that maybe looks like this, and it might have one flagella, it might have two flagella, maybe even three flagella. And the purpose of these flagella are to help to move that organism from place to place within the environment. Now, if you're a ciliate, you basically have a body surface that is covered in thousands of small cilia. And these small cilia are like little hairs. And so you would have an organism that might look like this, and its body is covered in lots and lots and lots of little hairs. And what's kind of different between the ciliates and the flagella is all of these cilia move somewhat in unison, and so that helps to give these particular types of protozoans a little bit more flexibility on how they move through the environment. Now, the third group are considered the amoebas, and what they do is they use something called Pseudopodia. Now, Pseudopodia is basically an extension of cytoplasm. Now, this extension of cytoplasm can come in lots and lots of different forms, and these are the forms that I'm expecting you guys to be familiar with. The first one would be considered a lobopodia. So this is more of a blunt, this should be tipped, not dipped, Pseudopodia, and so you would have something that looks somewhat like that. And so we have a blunt tip there. Now, Phylopodia would be thin and sharply pointed Pseudopodia, and so you might have something that looks like that. All right. Now, Rhizopodia would be somewhat of a branch type of filament. So if you think about a Christmas tree, you might have some Pseudopodia that take on maybe this type of form. And then you have something called Reticulopodia, and these are kind of unique because these actually have the ability to form sort of a net-like appearance. So you can see the distinction between the first four. Now, Axopodia is very similar to the Philopodia. If you notice, they're still thin, they're still somewhat pointed, but the difference being in that they actually have a central longitudinal axial filament of microtubules. And a microtubule is just a cell structure. So in addition to looking something like this, again, thin and pointed, they're also going to have a very rigid inside to them. All right, so that makes them a little bit different from the Philopodia. Now, there are some amoebas actually out there that form what we consider shells. Now, we give that shell a special name, and we call that shell a test. Now, this shell can be made of a silicous or a silicon type of material or a chitinous type of material, so they could be chitinoid. And if you recall, for some of you that may know a little bit about insects, they have what we consider an exoskeleton. And that exoskeleton oftentimes is made of a chitin material. Now, foraminifers and radiolarans both form tests. Now, as we had said before, one of the things that makes protozoans very important to the environment is they can form the substrate of oceans. Now, that substrate is formed by these tests. So when you have foraminifers or radiolarans that have decomposed in the environment, they become the ocean floor because they become basically the material that is primarily the test, the shell of that protozoan, since that becomes the floor of the ocean. So again, they're very important when it comes down to a lot of what you're going to see in the ocean environment. So moving on, we need to make sure that in regards to locomotion that we get a good understanding as to how Pseudopodia works. So we know the different types of Pseudopodia. So now we need to take a look at how does a Pseudopodia actually perform? How does it function? So before we do that, we need to distinguish between two different types of cytoplasm. Now remember, Pseudopodia are simply extensions of cytoplasm. But there's two types. We have something called an endoplasm and one called an ectoplasm. If it's an endoplasm, in terms of appearance, it's going to appear a little bit more granular, and it's going to contain the nucleus and all the cytoplasmic organelles. So it kind of forms the bulk of the cytoplasm within the cell. Now, what's interesting about this particular plasm is that it's a bit more fluid. So it has the ability to move through the organism a bit more freely. If it's an ectoplasm, then it does tend to appear a little bit more transparent. If it is found in maybe a flagellate or a ciliate, which of course they have flagella and cilia, it's going to be at the base of those structures, and it's often a little bit more rigid within the cell. Now down here at the bottom, you're going to notice we have sort of a cross-section through one of the pseudopods of an amoeba. So this is an amoeba in the upper left-hand corner, and they've taken a cross-section between this particular pseudopod of this example. And so that's what you see blown up here. Now I know this is kind of complicated to understand, but the only thing I really want you to get from this diagram is this. When you look in the middle of this pseudopod, you're going to notice we have what we consider flowing endoplasm. And that makes sense because, as we said before, the endoplasm is very fluid. So as this endoplasm makes its way towards the front of the pseudopod, you're going to notice we have something called a hyaline cap. Now this hyaline or hyaline cap is going to act sort of as a barrier for this endoplasm. So as it comes against this, this barrier, it's going to basically be deflected, and as it's deflected, it's going to make its way back into the organism. But as it makes its way back in, it's actually going to transform, and it's going to transform into a different type of plasm called ectoplasm. Now that's when the plasm becomes a little bit more rigid. And so as it passes through this point, you can sort of see this sort of net-like appearance, which is caused by various proteins and filaments coming together within the plasma itself. It's going to form this net-like structure, and it becomes very rigid. Now, as it pushes its way, again, continuously above that flowing endoplasm, that net-like appearance is going to sort of start to fall apart. So those proteins are going to start to break down. As it breaks down, it's going to become, again, a bit more fluid. So it's going to make its way back into the organisms itself. And if you notice at the bottom, it's happening exactly the same way. It's being deflected, and it's being moved through, becomes a bit more rigid, becomes ectoplasm, then eventually starts to break down, and again goes back into the organism's intercellular structure. So that's what I'd like you to know in terms of how Pseudopodia actually work. Some of these things you see here in terms of the proteins, the type of filament, etc. If you want to know those, that would be great, that would be super, but I want you to know the mechanics, what's actually going on, and where would you find each particular type of plasm at certain points within this process. So that's going to finish us up for our first screencast for Chapter 11. Please make sure that you fill out the form that is connected to the screencast, and we will see you next time.